Hi, everyone. I hope you can hear me. I'm Davide from the organizing committee. I will quickly remove my share and welcome to the next installment in our seminar series. Today we are discussing MRI. The topic is MRI and machine learning. We have with us Riccardo Dalbello from University Hospital Zurich. And the topic of today is a bit hot, so synthetic CT from MRI images for RT planning, radiotherapy planning. And well, the question is, the underlying question is, can we get rid of CTs for radiotherapy? Of course, it's a bit simplified and provocative. Before I let Riccardo speak, just one reminder, who needs a certificate of attendance, just leave the email in the chat and then we will send the certificates in the next days. Quick introduction of Riccardo. So he's a medical physicist from Italy. He studied in Trieste. Originally, his topic was diagnostic. He was working with breast imaging with synchrotron light. And then he switched to the other side of medical physics, that's radiotherapy. So he was in Germany, in Heidelberg, working with radiotherapy and ions. And then he moved to USZ to complete the training in medical physics. And now he has a 50-50 position, half clinical duties, half research. And his research focus is, of course, MRI only, workflow, and automation MRI and AI. So I hope I didn't forget anything, Riccardo.
Oh, that's a lot. Thanks a lot for the introduction.
Thank you. Just one comment. I hope everybody is seeing the slides. If not, please stand up, speak up. And then, yeah, Riccardo, you can start.
Okay, thank you, Davide, and welcome everybody to today's seminar. So the topic will be how to use synthetic CTs generated from MR data for what we call MR-only radiotherapy. And I'm aware that this is a seminar series in diagnostic imaging, so I will also take a little bit of time to introduce the topic of radiotherapy and why we need imaging in this context. So this will be the outline. So as I said, let's go ahead and talk about the motivation for synthetic CT in radiotherapy. I will discuss some commercial solutions for the state of the art, and then we will come also to the developments that we have been carrying on here at the USZ. And the general topic that we will discuss today is represented by the figure here on the right. So basically we have an MRI data set and we want to convert it to a CT or synthetic CT data set. But we also want to be extremely careful with what regards the quality of this conversion. So let's get started with the introduction. And this is the radiotherapy workflow, or as we know it nowadays. So the current workflow works as following. We always perform a CT scan of the patient, and we need a CT scan because we need to use this data to calculate the interaction between radiation and matter and to calculate the dose that we deliver to the patient or that we plan to deliver to the patient. In many cases, the CT scan might be sufficient, but nowadays it's more and more common to have additional imaging available, which could be MR data or even PET data that our doctors use in order to better define the target volume and the organs at risk surrounding it. And here you see the first problem that the current workflow has, that we are dealing with diagnostic MR scans or PET scans, and we need to register them to CT scans performed in treatment position. So I will show you some pictures. We have some mask systems, for example, to keep the patient in place. And of course, if the position is different between the two scans, we start introducing some uncertainties. Well, we perform our planning, and then the day of the treatment, we perform additional imaging that could be planar KV or a cone beam CT at our linacs, in order to reposition the patient to the best position possible, and also to monitor the patient during the treatment. So this is the current workflow. And in what is called the MR-only workflow, we basically would like to replace all the imaging modalities with MR imaging. And this includes both in the treatment planning and in the positioning and monitoring. So here I highlighted in green what is already available in the market and currently in clinical use. And you see that for positioning and monitoring, I already put this in green because there are the so-called MR Linux. USAID is operating one of these, where instead of using X-rays to position the patient every day, you have a MR integrated with a Linux for treatment positioning and monitoring. So the topic of today is this. I hear some background voices. Maybe participants, you can mute yourself. Thank you. So the topic of today is this red arrow, which is the conversion of MR data into electron density or CT, synthetic CT, which we need for those calculations, and also how to perform a quality assurance on this. But why do we want to move into MR-only radiotherapy? The three main motivations that are commonly reported in literature are the following: to reduce the radiation exposure to the patient during the planning, to eliminate the registration uncertainty, since we will now only have one image modality, and to be more cost effective, since we need to operate only one imaging devices. But what I think is also very important and often forgot is that we already perform MRI as standard of care for many cases. So if we can just use this data, then there is an intrinsic benefit. And yeah, so here are some examples of a prostate and head and neck case where you see the superior quality of MRI in defining the target structure and also the surrounding organ at risk. And here, for example, on the top right, you can see in the CT this mask system that we use for fixing the patient to a given position and be sure that also during the treatment, the patient stays in this position. And ideally, we will also like to perform MRIs in this setup to reduce the uncertainties. So the current workflow nowadays is this MR plus CT, which is well established. This is current technology and is current standard of care. However, there are some registration uncertainties and it's a bit cumbersome from time to time because maybe we need 2 appointments on 2 different days. The patient has to come twice to prepare to start preparing the treatment. Instead, we want to switch to this MR-only workflow, which, however, requires some developments in synthetic CT. So, and these developments, so they come from the realm of artificial intelligence and neural networks in particular. And I like often to present this graph where you can see the hype cycle for artificial intelligence. And if you check where we were in 2022, you can see that synthetic data is at the peak of inflated expectations. So everybody is expecting that maybe synthetic CTs will totally remove real CTs from the radiotherapy planning. But let's see how will it go in the next years. So what I think is important to keep in mind is also the understanding of what's going on, how to, and this is important for QA, because if we have a lot of data, and this is needed to train our networks, we can basically live in a world without physics. So we have a lot of data, we train our network, and we get our synthetic CT out of it. And this is very good for automation. So we can perform this process in the background without much human interaction. However, we also want to perform quality assurance. And for this, I think it's very important to have an understanding of the single steps of what's going on. And maybe we have a bit less data, but we can troubleshoot in case we see some differences from our expectation. So let's go to the state of the art and let's check what are the commercial solutions available to perform this conversion from MR to synthetic CT. And first, a historical picture. So the use of neural networks for this task has been proposed in 2017 by a paper by Han et al., where they firstly investigated the conversion of MR data of the brain. And you can see that ever since this first proposal, this topic has been exponentially growing. So this data stops 2 years ago, but if you search on PubMed, this topic is exponentially growing. And most of the data comes from high field images or above 1.5 Tesla, and is using this UNet or GAN architectures, which require pair data. And not surprisingly, brain is one of the most interesting sites to look at, and the first ones that have been investigated, because due to this max fixation system, you can have the same positioning in multiple imaging modalities, and also the brain is a rigid structure, which if you didn't have the mask system in DMR, you can register to your CT. But also head and neck and pelvis have been hot topics and have been investigated, and commercial solutions are available. We see a fewer number of investigations for thorax, lungs irradiations, for example, and in the abdomen, mostly because of the moving anatomy and the lower quality of the imaging. So there are several companies in the market providing solutions for this. And again, here, as you can see, brain and pelvis are somehow state of the art, and they are clinically available, commercially available with certified product. Head and neck, there is one vendor which has it available, but due to data scarcity, this is also ramping up, and there have been quite some developments also in this field. And then abdomen, breast, thorax are still under investigation. So before looking at some of the images and some of the data, I'd like to show how this conversion is performed. And to do so, I will use as an example one specific network, the CycleGAN, which we have been using in our studies. And I think is also pretty easy to understand what's going on. So the advantage of CycleGAN compared to other architecture is that it can be used also for unpaired data. And this is very important in the abdomen, where you have physiological changes. And basically, you have stomach and bowel feeling that are different between the MR and the CT scan. So your images are intrinsically not paired images. They are showing you different anatomies. So in this case, we can use MR and CTs from same patients, but they don't need to be registered. And we have several ingredients or components that we need to use to perform this conversion. So the first ones are generators, are functions that take one image from one domain and transform it as a matrix in an image in another domain. And we need two generators, one from transforming MRs into CTs, and the other one CTs into MRs. Then we need discriminators to basically decide or evaluate whether the image we are looking at is a CT or an MR. And then we have to quantify the difference between the real and the generated image. And to do so, we do it in a cycle. So we perform the following. We start with an MR, we convert it into a synthetic CT, and then we back convert it into a synthetic MR. And if everything is done correctly, basically going from one domain and to the other and back to the original domain, we should regenerate the same MR that we started with. If we don't, we have a difference between the images, and here is where we put our loss. So we can try to optimize the network by reducing this cycle consistency loss. And then we do the same starting from the CT, so generate synthetic MR and then back to a synthetic CT. And so this network is robust. And then, of course, we train so many different components of this network. The only element that we are using at the end is this function G to transform MRs into CTs. Very well. So once we have our network trained and ready to transform data, we have to consider how to implement this into a clinical workflow. And there has been a paper published this year actually proposing a stepwise approach with 5 phases in order to switch from the classical workflow that I discussed at the beginning to the MR-only workflow. And all the intermediate steps are some intermediate workflows done during commissioning, evaluation, and fine-tuning maybe of your network. So the classical workflow is what I've discussed earlier. Phase 2 is basically using MRs into planning position and not anymore in the diagnostic setup. And this is something that we have been doing at USZ since 2019. So we acquired quite some experience with this MR-Linac, this hybrid device, where patients are imaged and treated in the same device, and therefore they also have a fixation system like masks for head and the neck, or these vacuum pillows for other locations. And you can see that we have treated several cases from brain to prostate, head and neck, even in the thorax region. So phase 3 then is introducing in the workflow the synthetic CT, or here in this image also called pseudo CT. So what you do in phase 3, you do your planning MR in the treatment setup, so with a flat bed top and the fixation system. You still acquire a planning CT, and you fuse these two, so as you would normally do, and you perform your planning on the planning CT. However, you also generate a synthetic CT out of the MR data, and you can retrospectively evaluate how well your synthetic CT is compared to the real CT. And this is what we have also been doing in several studies with networks that we have developed here at USZ in order to retrospectively evaluate how well your network performs. And in general, you can see here some reports from Erlangen and from the Harvard Medical School. Normally, those deviations of the relevant targets and organs at risk are within 1%. So at the end, we are interested not only in having good images, but also having those calculated accurately on such images. Phase 4 is then what is ongoing right now at the USZ. In our department, we are performing our planning MR, we are generating our synthetic CT, and we are performing the planning on the synthetic CT, not on the real CT, but still in the background, we are acquiring a CT in order to compare the two results and be sure that our synthetic CT is accurate enough for our needs. And accurate enough for us has two meanings. The first one is for dose calculation. We want to make sure that if we calculate dose on the synthetic CT, we get the same dose that we will get on the CT. And we can quantify this also with some other simplified approaches, what I mentioned before, with a bit less data and more physics into the context, and I will come back to this later. But also for us, it's very important also the matching. So then when we go and perform the treatment, we perform our KV-KV or COM-beam CT to position the patient, and we want to make sure that the registration MR to CT or MR to synthetic CT, and then including also the COM-beam CT in this triangle, delivers accurate results. So there is a twofold evaluation on those and on the matching quality. And then phase 5, once everything works properly, one can go into MR-only planning and remove the CT from the treatment planning workflow. And this is what we have already done for male pelvis. So we are nowadays under certain inclusion criteria, treating prostate without performing a CT acquisition before the treatment delivery. So we perform only MR imaging, and we have observed that the dosimetry is good, is within 1%, and also the preliminary evaluation of the matching is within our requirements. And that's why we went into this. We will do it also for female pelvis, and for brain, the investigation is still ongoing. So what do we do practically? So we have a solution by Siemens. And what we do is that when the patient comes, we acquire the diagnostic images that are required for contouring. And then on top of this, we also acquire a T1 Dixon, which has a large field of view, has distortion correction, and this is used to generate the synthetic CTs. Of course, one can always add more sequences into the protocol in order to support contouring. And then the results that we get are the following. For brain on the left, for pelvis on the right, you see the Dixon, and then we have the generated synthetic CT, where you can see the classic contrast that one will get also from the CT, however, with a bit more blurring of the bones, since this is generated from MR data. And the time requirements at the 1.5 Tesla scanner that we have in our department range from 2 to 4 minutes. So the additional time required to acquire this Dixon sequence is in the order of 3 minutes. So as a summary for the clinical part, or let's say the state of the art, there are site-specific solutions that are available and certified. The time requirements at the MR scanner is increased by 2 to 3 minutes with the patient already in the scanner. And the dosimetric and matching performances are good, but every center that wants to implement such a solution has to prospectively evaluate this. However, quality assurance solutions are currently not provided by the vendors, and therefore we have looked into what one could do, and the idea is to perform an additional calculation on a different electron density map that could be extremely naive. So we could just say, okay, our patient is water, let's see what is the dose there. Or there have been some reports here on the right where people developed a patient-specific phantom out of the MR, and basically you can get to those deviations smaller than 1%. Of course, we want to be somewhere in between for the clinical workflow, and this is also what we will discuss later on. So now I will quickly go over some developments that we have had here at the US side. And this is twofold. On one side, the generation of synthetic CT for sites which are not currently standard of care, such as abdomen or head and neck. And the other part is the quality assurance. So first of all, for the abdomen, we used this network, the cycle gun, which I discussed earlier. And out of this MR LINAC, we acquired retrospectively data from almost 200 patients, so 186. And these are low field images. So the MR that we have in our scanner combined with the LINAC is a 0.35 Tesla MR. And yes, and these are the features of the MR data that we acquired. So some images that I hope that speak more than words. So on the top, you see the MR that we started with, and at the bottom, you see the CT acquired with the patient lying on the same setup, but within half an hour. Usually within half an hour, we acquire the CT. And you can see that the air pockets in the stomach, they move quite significantly. So these images are not matched to each other. They are not paired data sets because of these physiological movements. And that's why we use this cycle gun architecture, because it can compensate for this. And as a matter of fact, when you then generate your synthetic CT out of the MR data, you can see that the air pockets are reproduced correctly. And I'm not discussing air pockets just because they are nicely visible, but also because they are highly relevant for us for those calculations, because the radiation that we deliver is not attenuated through air, and therefore if there is or if there is not air, this plays a difference in the dose delivered to the target and to the organs at risk. Therefore, we have to take this greatly into consideration. So here, another case where you can see the CT in the lung window. Also, the lung tissue is nicely reproduced for this treatment of the liver. But at the end, we are not interested only in image quality, but also in differences of those delivered and calculated on the two images, so the synthetic CT or the planning CT. And here I reported the dose difference, the relative dose difference for several structures. So PTV and GTVs are our targets, and then some organs at risk in the region. And you can see that the mean dose differences are well within 0.5%, with max deviations limited to 2%. So this was the case for the abdomen. Then for head and neck, we decided to use a slightly different network, this residual visual transformer. And this has been also recently published. I think the paper was accepted only last week. So this is also a preview. And also in this case, we used low field images. And we trained the network in a pseudo 3D approach where we didn't feed all the 3D volume in one go, but we basically fed the 3 orthogonal planes as 3 different input channels of the network and to then generate our output. And also here you can see the differences between the original MR and the synthetic CT here on the right. They are very similar. So also in this case, for those calculations, we hardly see any differences. And therefore, we also wanted to look at something else, which is how to contour or automatically contour structures on such images. And the reason is that we also use neural networks in order to smoothen and speed up our workflow such that we do not have to manually contour every single structure that we have in our treatment plan, but we start with some automatic contouring performed by a software. In this case, it's a commercial solution, MIM, where we can feed a CT into the software, and we get out the contours of some structures such as oral cavity, salivary glands, parotids, and so on. And the idea is that if we go into an MR-only workflow, all these auto-contouring softwares, they have been developed to be used on CT data. But if we feed synthetic CTs, which have a bit lower quality, are we getting there? Are we achieving the same quality of the contours that we get out? And the answer in this case was yes. So you can see here for the oral cavity, for example, the differences between the auto contours on CT and synthetic CTs are very similar if then are compared to what our doctors accept at the end. And if we compare synthetic CT to CT itself, we hardly see any difference. So here is the DICE score coefficient, for example. Then the last topic that we investigated is the quality assurance, how to perform quality assurance of this synthetic CT that we generate. And I would like to start with saying that there are, at the moment, no national and international guidelines on this topic. So there are several working groups at a European level, at a Swiss level, in order to start with the state-of-the-art surveys and so on, as you can imagine, in order to go towards guidelines. But these are incremental steps, and we are not there yet. what happens is that in the review papers or in novel papers with new science, you see basically a zoo of parameters that one can use to evaluate the image similarities. So we have over 15 to 20 parameters that one can evaluate, but then for clinical use, one would like to simplify all of this. And the idea is that once we go into MR-only workflow, we do not have anymore this CT in order to retrospectively evaluate what's going on and evaluate the quality. So one has to find different solutions. And one elegant solution, I think, is that when we use cone-beam CT for positioning the patient, one can recalculate the dose on a corrected cone-beam CT, because at the end, this is CT. And it has been shown that for prostate, one can achieve those deviations within 1%. So this is one possibility. The other possibility is more important when you go into MR-Linac treatments where you do not have this CT. And as I said earlier, you have a range of different complexities that you can use in order to test your synthetic CT. And what we try to do, we tried to start it from a very naive representation of the patient with water to go into bulk densities that you can automatically or manually contour. So you say, okay, there are 5 tissue classes, which I know the density of. Let's have a model of the patient based on this. Or you can have, for example, a second independent neural network trained on a different set of patients. And of course, as you increase this complexity, you also expect a better precision of your quality assurance. And this is also what we observed. And we investigated this for standard cases where there are these massive air pockets that I showed you earlier, where there are lungs along the beam path, or even with implants. And you can see that if you use this independent neural network, so the orange bars here, if you span over all different cases, you are always within the 1 to 2% deviation. Outliers never go out of the 2% bars. So I think I will skip this because we are in interest of time. And I will just show you an extreme case of what happens when we generate synthetic CTs of patients with implants. What happens is that most of the time, these networks are trained on data that does not have artificial implants within the training data set. So as you can imagine, in the MR, we do not see the implant. It is not present in our training data. And therefore, in the synthetic CT, this implant just disappears. And instead, if we perform a CT simulation of the patient, the implant is clearly there. And in this case, it was even within the target structure. And if you compute the dose differences, you can see that you can underdose or overdose this local region. And this is one of the reasons why for enrolling a patient into MR-only workflow, it's very important to screen the patients and be within the limits of the training data set of your network. Otherwise, you have cases such as this one, which are clearly an exclusion criteria, and a CT is needed. So to conclude, I showed you that there are site-specific commercial solutions that are available and can be implemented in the clinic. However, guidelines are not available yet on how to do this, and therefore, some work is ongoing on defining all the quality assurance criteria. Synthetic CTs, the image quality is a bit lower compared to the normal planning CTs. However, they are suitable for dosimetry and for matching and patient positioning. And quality assurance is one of the most important topics that I think needs to be investigated, since end-to-end testing is not possible. We always need a CT of the patient to perform this end-to-end testing. With software solutions, we can be within 1 to 2%, and we have to be sure that we do not increase the total uncertainty budget of our radiotherapy workflow by using the synthetic CT. And then, of course, the complete chain of quality assurance includes also other factors such as imaging QA, data transfer, and so on, which I didn't discuss in this presentation. So with this, I'm done, and thank you for the attention, and I'm happy to take questions.
Thank you very much, Riccardo. Very, very interesting. Yeah, we have 9 minutes for questions, so please. Just unmute yourself and speak up. Or in the meantime, Riccardo, I can give you a comment. So I was a bit surprised that bones seem not to be a problem. So I would have expected otherwise. You said that there is some blurriness on the bone structures, of course, because you start from an MRI image, but in the end you have dosimetry with sufficient precision. So
yes, so this is a very relevant comment, actually. So if we, here are some images generated by our in-house developed or trained network. And you can see also, for example, in the coronal images here, how the ribs are a bit blurred, whereas on the CT, they are nice and sharp. So for radiation attenuation, what matters is the total water equivalent path length. So radiation usually comes orthogonal to the axial plane. So is the total water equivalent path length until the point where you want to calculate radiation. And we usually deliver radiation from a 360 degree or from 360 degree angles around the patient. And therefore, if the bone, the ribs in particular, are not so nicely reproduced or they are a bit blurred, what this does not compromise much the dosimetric accuracy of the synthetic CT. So for those calculations, even if you have unsharp bones, but they have the correct Hounsfield unit values along the beam path, this is totally fine. However, for the matching, this is more problematic because when we do the matching with our COM-beam CT is a bit more noisy, is a bit more, has a smaller field of view, and we really rely on the bones for matching. So if you have to match your COM-beam CT to a synthetic CT which has blurred bones, then you have a problem. And then the matching quality decreases. However, this problem is somehow removed at the MR LINAC because we do MR to MR matching and the synthetic CT is in the background only for those calculations. At the normal LINAC, yes, this can be a problem and has to be evaluated. So the blurriness of the bones is more relevant for patient positioning rather than those calculations, because with photons, we are pretty robust against such small differences. If one uses protons or electrons, then as your radiation, then this is a different story. But yeah, this was not the topic of our investigation. But yes, good point.
Thanks for clarifying that. We have room for other questions.
Hi, Riccardo. Thank you for the great talk. I just have a kind of a question, or I'd like to ask for your comment on kind of the amount of data that you used for the training. I think you mentioned somewhere it was like less than 200, which seems quite low nowadays in machine learning. So is there like a minimum amount of data that you use, or yeah, can you just comment a bit more on how much data ideally you would ultimately want for this?
Yes, yeah, that's a good point. So these networks are designed to be used with millions of images as training data. This, of course, is something that we do not have available in our retrospective databases. So for the abdomen, we use this cycle gun, which is unpaired training, and this is a bit more demanding. And that's why we used all the patients that we had, whole order of 200 patients, but is many more images, because every patient has a 3D volume that is imaged. And the field of view is in the order of 40 centimeters, and we have a slice thickness of 2 to 3 millimeters. So every patient delivers more than one image. So you have to scale up this. However, if we go to the head and neck case, you see that we only had 31 patients available because we do not have many patients in this study. And that's why we used a different network, which used paired images. So you basically remove from the network this need of taking care of having unpaired images, but you know that every voxel in the CT corresponds to a voxel in the MR. This can create some problems if the images are not perfectly matched. However, this compensates the fact that we do not have so much data available. And there have been some studies reporting that, of course, there is a trend. As you increase your amount of data, you also improve the output. But it saturates pretty quickly. So if you are already in the order of 20 to 30 patients with a 3D volume available and good quality data, this is also important, you do not get much better results than having order of 200 patients with bad quality data. So it's a bit more important filtering your data at the beginning and be sure that the images are correctly matched, you do not have implants, you do not have contrast media, and so on. And then you can also work with such numbers, 30 patients or so. But in principle, the more, the better. Yeah.
Cool, thank you.
Okay, 2 more minutes. Anybody has questions? If not, before closing, I have maybe one comment for Ricardo. You mentioned head, neck, and pelvis. What next?
Yes.
If you can say.
So if I go back to the slide at the beginning here, I think one should also check what is clinically relevant. So there have been some studies for breast. You can generate synthetic CTs for breast, but we don't use MR data for contouring the breast. So I think abdomen is the next site where this could be extremely useful because you can see the lymph nodes very well for them preparing the radiotherapy plan. Thorax, for lung irradiations, honestly, CT for these CTs are performing extremely well. So I don't see thorax coming anytime soon as a hot topic. I think the next one will be abdomen that will go into the CE certification and an introduction in the clinical workflow.
Okay. We're looking forward to that. Maybe next year we might have an update on.
Yes.
Okay, and instead, final remark before going to lunch. Next month, we have on the 17th of August, another talk on vision language models in thoracic imaging. So the body region was kind of connecting last question to the next topic. So looking forward to have all of you next month connected. And small anticipation, we plan to make all the recordings available at some point on some platform, probably the hospital website. So I will tell you more next time. Okay, thank you again, Ricardo. Thank you all for participating, and talk to you soon.